Hey everybody, this is the Krebs cycle made easy. I am Dr. Sean the Magic Mon. Please follow me on Instagram. Before we talk about the Krebs cycle, I need to tell you where we get the carbons from that we put into the Krebs cycle. We get those carbons from glucose. Glucose comes from carbs. We digest carbs, break it down to the smallest component called a monosaccharide, and we put that into the cell. So here is their cell. Glucose has 6 carbons. Insulin is important to bind to the gate, I would say. It's like the key. Facilitated diffusion opens the gate and glucose goes into the cell. Here is 6 carbons in your cytoplasm. Now we need to get that 6 carbon glucose and put it into the mitochondria, which is the motor or the powerhouse of the cell. Now 6 carbons is too big to fit. We need to cut that in half. We need to glycolyse it. Glycolysis. So glycolysis is the process of cutting the glucose in half, and now we have pyruvate. Pyruvate is driven in the mitochondria. It's driven in with carnitine. I use a mnemonic, you drive the car in. Carnitine drives the carbons in, and the carbons will be a 3-carbon pyruvate, which will go into the Krebs cycle. There's 2 ways to get into the Krebs cycle. I should say there's one way to get in there, another way to create one. But the Krebs cycle lives in the mitochondrial matrix. Its job is to make hydrogens. And hydrogens go to the inner mitochondrial membrane where the electron transport chain makes ATP. So the Krebs cycle has 10 steps. These substrates are what we call ketoacids. A keto acid has the property of a sugar and a property of an amino acid together. So amino acids could go into the Krebs cycle, and so can sugars. My mnemonic in order is our cousin Cecilia is oversexed and keeps 665 men. That's kind of funny. All families have a cousin Cecilia in it. And if you're not laughing, that's probably you. I'm kidding. Our stands for oxaloacetate. Cousin stands for citrate. Cecilia, cis-aconitate, is isocitrate. Oversexed is oxalosuccinate. And keeps alpha ketoglutarate. 600 is succinyl-CoA. 60 is succinate. 5 is fumarate. Men is malate. Our cousin Cecilia is oversexed and keeps 665 men. So think of these 10 substrates here as intermediates. That would be like if I want to fly from Kansas City to Los Angeles, there's always a stop. One stop in Denver. These substrates would be a change planes, not a layover. But when you change planes in Denver, be careful of the Illuminati. So we have all these steps here. Let's go over this. Our is oxaloacetate. Cousin is citrate. Cecilia cis-aconitate is isocitrate. Oversexed is oxalosuccinate. And keeps is alpha-ketoglutarate. 600 is succinyl-CoA. 60 is succinate. 5 is fumarate. Men is malate. So the easiest way to get into the Krebs cycle is via acetyl-CoA. Acetyl-CoA has 2 carbons. Oxaloacetate has 4. 4 plus 2 is citrate, which is 6. And it goes around 6, 6, 6, 6. You lose a carbon 5. You lose a carbon 4. 4, 4, 4, 4, 4 plus 2 is 6, 6, 6, 6, 5, 4, 4, 4, 4, 4, 4 plus 2 is 6, and so on and so on and so on. There's another way to get into the Krebs cycle. Pyruvate. Pyruvate has 3 carbons. You can carboxylate, meaning add a carbon to it, to get oxaloacetate. That's called gluconeogenesis. And now when you have oxaloacetate, you grab another acetyl-CoA to get citrate. So why would our body want to carboxylate pyruvate? To create a new Krebs cycle? Now you just doubled the amount of ATPs you've created. So let's go over the energetics from the Krebs cycle. When I say energetics, how many ATPs do we make? If you see between isocitrate and oxalosuccinate, we make an NADH. Between alpha-ketoglutarate and succinyl-CoA, we make another NADH. Between malate and oxaloacetate, we make another NADH. So NADH, when it goes to the electron transport chain, makes 3 ATPs. Remember, we have 3 of them. So that's going to be 9. We have an FADH2 between succinyl-CoA and fumarate. FADH2 makes 2 ATPs. And now we have a GTP between succinyl-CoA and succinate. GTP is ATP. It's just a different currency. If you want to say Canada has its dollar, America has its dollar, they're both dollars, different currency. So GTP makes ATP at the substrate, which is the substrate level is the Krebs cycle. That's 1. So 9 plus 2 plus 1 is 12. We make a total of 12 ATPs in the electron transport chain from the Krebs cycle. So the majority of ATP is made in the Krebs cycle. So really quick, substrate level phosphorylation means you make ATP at the substrate level, which is the citric acid or Krebs cycle. The rest of them go to the electron transport chain to make ATP, and that's via oxidative phosphorylation. So there you go. That's the Krebs cycle. I'll have other conversations about glycolysis and the electron transport chain later. I hope you got something out of this, and I hope you're going to use these mnemonics to help you pass the test.